We're going to be covering Chapter 4 in today's lecture, where we're learning about the mechanisms behind classical conditioning. So this is going to be pretty much a direct continuation from last chapter where we talked about the general principles of classical conditioning. Now we're just going to dig a little bit deeper. So as usual, let's start with an outline. We're going to cover what makes an effective conditioned and unconditioned stimulus, what determines the nature of our conditioned response, and then we'll talk about how do the CS and US become associated. For this lecture, we're probably only going to get to the first point, possibly the second point as well. We'll see how our time is doing, but we'll finish up the rest of the chapter for Thursday's class. So some things that we want to keep in mind or consider when we're looking at conditioning is the idea that conditioning can depend on our prior experience with each of our stimuli, whether we have seen our CS or our US before the conditioning setup. It also matters how relevant the CS and US are to each other. It's a lot easier to use a CS that belongs or fits with a particular US. The presence of other stimuli during a conditioning trial can also affect how that conditioning proceeds. And one last thing to keep in mind is that not all conditioning is reliant on a direct CS-US pairing. As a slight hint of what's to come, we can talk about higher order conditioning where we can remove having an unconditioned stimulus directly present because we're just associating a new CS with an existing CS. But we'll get to that later on in the lecture. So let's start by talking about what makes a good conditioned stimulus and a good unconditioned stimulus. The first thing we want to talk about is knowing where those stand before we begin our experiment. So if you're trying to set up a classical conditioning experiment, you need to know what is your CS and what is your US, and where are they sitting? What do we know about them? How are we going to treat them right off the bat? The nature of that stimulus might determine that in some cases it serves as a CS, and in other cases it could be a US. The example that the textbook uses is that the flavoring sucrose can serve as a conditioned stimulus in something like a conditioned taste aversion test, where you flavor a food pellet with sucrose and then induce nausea, which would then create an aversion to that conditioned stimulus, the taste of sucrose. But if you use sucrose as a food reward, or just as a food in general, in a sign tracking experiment, then it can become an unconditioned stimulus, where we have a natural response to sucrose as a type of food. So based on the framing and based on what that particular stimulus is, we can call it a CS or a US depending on that situation. Continuing on from that point, we can talk about the novelty of a stimulus. Basically, how much experience do we have with the stimulus that's being used, whether that be our conditioned stimulus or our unconditioned stimulus. And the general rule of thumb here is that the more experience that we have with a particular stimulus, the slower the association is going to be, or the slower learning will occur. If we have more novel stimuli, if it's something that you haven't been pre-exposed to, if it's something you don't have a lot of experience with, then the learning will happen quite a bit more quickly. So let's talk about our conditioned stimulus first. This is called latent inhibition, or conditioned stimulus pre-exposure, where you basically have repeated exposure to the conditioned stimulus before that conditioned stimulus is ever used in conditioning trials. So if we were looking at Pavlov's dogs, where we have a tone paired with meat and it leads to them salivating, then our conditioned stimulus is that tone. And before we started pairing the tone with the presentation of food, we would then just play the tone over and over and over again to the dogs without pairing it with meat. So that pre-exposure is happening before the classical conditioning would happen. And it ends up actually leading to habituation. Basically, our senses try and limit the processing and attention that we pay to those repeated stimuli. especially when those are inconsequential stimuli that aren't really telling us anything. A neutral stimulus, because at that time, the tone doesn't mean anything to the dog, so it is neutral. It's not giving us any extra information, and so it can be safely ignored. At least that's what our brains think. So this latent inhibition process is going to tell our brains that we can ignore that conditioned stimulus even when it becomes associated with something later on. And so that leads to much slower learning. Next, we can talk about our unconditioned stimulus. And so we have our unconditioned stimulus pre-exposure effect. And as you might have guessed, it's caused by repeated exposure to our unconditioned stimulus before that unconditioned stimulus is used in conditioning trials. So this diagram here goes with our unconditioned stimulus pre-exposure effect. And so in this setup, we're looking at rats in an auto-shaping setup. So in our pre-exposure, they would be exposed to either chocolate or bacon. So you'd have one group that was exposed to chocolate and would become pre-exposed or familiar with chocolate. The other group would be pre-exposed to and become familiar with bacon, because we want to look at more than just whether they like chocolate or like bacon more. Then we would go into our Pavlovian conditioning trials, which in this case would be an auto-shaping procedure, where they would insert a lever into a chamber, and then after the presentation of the lever, the rats would be provided with food. They would be given either bacon or chocolate, sort of intermixed. They would be different kinds of trials so that rats would get exposure to both. And then our measure of responding is how much they press or interact with that lever. So on our graph here, we have our y-axis is responses per trial, how much they interact with that lever. And then across the bottom, we have trials, which is our time measurement here. And so we can look at the two different kinds of conditioning trials, whether they were exposed to a novel food or a familiar food. So if they had been pre-exposed to chocolate and they were then given a chocolate conditioning trial, that would be a familiar food. But if they had been pre-exposed to chocolate and were given a bacon conditioning trial, then that would be a novel food. And so for our amount of responding, we see that when they're given the familiar food, the rate of responding stays pretty much consistent. There isn't a dramatic increase in responding. There is a bit of up and down here, but that's kind of what animal data tends to look like. What we care about is the fact that our other line, the one with the open circles and the solid line that's related to our novel food, we do see more responding over time in trials from novel stimuli. So we're basically seeing that there was learning happening. They've acquired this association, and they acquired it much faster when a novel food was used as their unconditioned stimulus. And what we think is going on here is that there's some kind of associative interference of the contextual cues present when our unconditioned stimulus is administered before our conditioning trials. Basically, pre-exposure to an unconditioned stimulus causes some associative interference with that particular stimulus, but if we have a novel stimulus, then there isn't any of that interference, so they learn much faster. The next thing that we should consider about our stimuli is their salience. Now, salience is kind of a difficult word to define, especially in terms of learning, but basically we use it to mean something like significance or noticeability. So a more salient signal is going to be something that you notice a lot easier. And as you might expect, a more noticeable or a more salient stimulus would probably make for better learning. And when we talk about salience, we can talk about the intensity, the biological relevance, or other qualities of that stimulus. We can start with intensity because that's a pretty simple one to conceptualize. Basically, the more attention-grabbing a stimulus is, the easier it is to learn something about it. So if we were using a tone, A louder tone would be a more salient stimulus. A brighter light would be a more salient stimulus. A larger stimulus would be more salient. And this salience can have an effect on both the unconditioned stimulus and the conditioned stimulus. So we can care about the salience of either our conditioned or unconditioned stimuli. So it's a lot easier for a dog to associate a really loud tone with a really good tasting food than it is for them to associate a very quiet tone with a very bland food. So we'd expect in that first example to see a lot of salivation very quickly, so lots of learning very quickly. And then in the other situation, it might take more pairings, more trials to see an association form. For biological relevance, we can look at that relevance to an animal's environment or to its current needs. We'll start with the environment, which is basically saying things that are relevant to what they'd encounter in their natural environment. So males may pay attention to a conditioned stimulus because it looks like a female. And so if we actually hop to the next slide here, we can have a more naturalistic stimulus. This stimulus here looks more like a female quail than this stimulus here. By adding extra characteristics that make it look like a female, they can make it more salient because it's more naturalistic. I think we've mentioned quail before. Quail are often used in studies of sexual conditioning because they will approach a model like this and start engaging in copulatory behaviors. And the more this stimulus looks like a female, the more intense those behaviors will become. We hop back here, we can also look at biological relevance as relates to an animal's current needs, and we can actually create a sort of an imbalance that makes a particular stimulus more relevant or more appealing so that it's more salient. A really simple example of this is that in a lot of studies that we'll look at, animals are moderately deprived of either food or water. Basically, you would test them before their mealtime so that you know that they're hungry, so that they'll do the behavior or be more motivated or be more reinforced when they get the thing that they need. This also applies to classical conditioning as well, where if you have, say, an organism that is salt deprived and our unconditioned stimulus is salt, then we might see faster learning because it's a more relevant and therefore more salient stimulus. So in all of these cases, the more intense, the more relevant, the more salient your stimulus, the faster we see for our learning. Now those points have applied to the conditioned stimulus and the unconditioned stimulus when considered separately. Now we can also consider our association or relatedness between the conditioned stimulus and unconditioned stimulus together. So we can call this our CSUS relevance or belongingness, and it's basically saying, do these two stimuli go together naturally? Is this something that we would associate together naturally? And if they match, it's a lot easier to learn an association than if they don't match. Now, the standard example to illustrate this is research done back in the 60s by Garcia and Kelling. I'm really bad at pronouncing names, so possibly Kelling. So this is a really, really cool experiment. You may have seen this multiple times before just because this is the experiment for belongingness and relevance, but whatever. We're going to go through it again just because it is that cool. So in this experiment, our subjects are rats, and we're looking at how their exposure or conditioning with a compound stimulus leads to associations with different kinds of unconditioned stimuli. So in this case, our compound stimulus is a taste and an audio-visual stimulus. What they basically have is a water bottle, and the water is flavored. The flavor could be either salty or sweet, And whenever they licked their water bottle, there would be a brief audio-visual cue where there's sort of a clicking sound and a bright light flashes. So we have this compound stimulus where we have both a taste, taste about sums it up, and then as well as a more external measure of auditory and visual stimuli. Now, what they did was they'd have two different groups. One group that would be exposed to this conditioned stimulus, this compound conditioned stimulus, and it would then be followed up by an unconditioned stimulus of a shock. That was group 1. Group 2 would get the same compound stimulus, and their unconditioned stimulus instead would be sickness. So the shock would be a mild shock delivered through the floor, or the sickness would be nausea that was induced due to exposure to some kind of chemical. Now, the cool part here is that both the shock and the sickness are aversive, unconditioned stimuli, and both would have a particular behavior associated with them. So sickness would be something where you don't want to drink that anymore because it made you sick in the past. It's a conditioned taste aversion. A shock would induce a fear response, which would also lead to reduced drinking of water because you're freezing and not doing a response. So in both cases, we would see if you learned to associate a particular stimulus with either shock or sickness, both cases you'd see a reduced responding or reduced licking of water. But what we want to do next is in our test trials, we can pick apart both of the pieces of our compound stimulus. So we could look at the taste and that audio-visual cue separately. So for our test trials, the rats would be exposed to either sweet or salty water, so just tasty water, or they would be exposed to noisy, bright water, where there would be that auditory cue and a visual cue, but not together anymore. And then we test, okay, do we see that reduction in licking due to taste, or is it due to audio-visual stimuli? Which, if we look at our next slide here, we see that we actually have, it differs or it depends on what their unconditioned stimulus was. So for those rats that were made to feel ill, the rats that got sick, they reduced their responding, but only to the tasty water. So they would still drink quite a bit to water that was associated or presented with bright lights and loud sound. They didn't mind that water. But if the water tasted sweet, they would avoid it because they had learned that the sickness goes with taste. And on the other side, the group that had received a shock, they would respond just fine and they would drink the tasty water with no problem, but they would show much reduced responding when there was an audio-visual stimulus. So the audio-visual stimulus goes with a shock. For logic, this kind of makes sense. Things that we eat are what we are hardwired to associate with what makes us sick. So if we have a conditioned taste aversion, it's us associating illness with a particular taste. So it makes sense that they would associate taste and sickness. And then if we think about audio-visual stimuli, that's stuff external to us. That's stuff in our environment. And a shock is something painful coming from the environment. So it makes sense logically to pair those two as well. And this gives us that idea of belongingness, where things that you eat go with or belong with being sick, and things in your environment go with other things from your environment. And so we can apply this where we would see more rapid learning, this decrease in responding when those stimuli match. So you can keep this in mind when you set up a classical conditioning experiment, because if you make sure that your CS and your US match, that they belong together, you can see much faster or much stronger learning. Next, we're going to get into that idea of learning without an unconditioned stimulus. Now, I already kind of dropped the hint that this would have to do with things like higher order conditioning. We're also going to look at sensory preconditioning, and we'll talk about both processes at more detail. But if we think logically, classical conditioning or Pavlovian conditioning would be fairly limited and of very little interest to us if we were stuck or constricted to only direct exposure to an unconditioned stimulus. Basically, we would only learn about things that were directly paired with food or sex or some other kind of biologically relevant stimulus. But in the real world, we see so many other associations. It's not just unconditioned stimuli. We also form associations with other conditioned stimuli. And that's where this learning without an unconditioned stimulus comes in. So we can say that our Pavlovian conditioning can also occur in situations without an unconditioned stimulus. So we can have our higher order conditioning and we can have our sensory preconditioning. So let's look at our higher order conditioning first, because this is one that you've probably already got quite a bit of experience with, so it should be fairly straightforward. There are two phases associated with higher order conditioning. Phase one is that we need to have an initial conditioned stimulus that is associated with an unconditioned stimulus. So we do need that unconditioned stimulus to get everything started. But once we've trained up this conditioned stimulus to produce a conditioned response, we no longer need that unconditioned stimulus to be present. Once we've gone through step one, we can move on to step two, where we take a new stimulus and we associate that new stimulus with our already trained conditioned stimulus. So now we have a secondary conditioned stimulus that we associate with our first conditioned stimulus, and that new CS will then come to produce a CR. We can also see much higher levels. This here would be an example of second order conditioning because we have two separate steps, but you can see third order conditioning or even higher. Just keep in mind that the further removed you are from the unconditioned stimulus, usually the less strong of a conditioned response that you see produced. That's not always the case, but a general rule of thumb. If we wanted to see this in action, we can look at the acquisition of irrational fears. The textbook uses the example of a woman who had previously been injured in a crowd. So in that step one, our conditioned stimulus is a crowd. Our unconditioned stimulus would be injury, because sometimes maybe you get bumped into or somebody steps on your foot or you get jostled around in the crowd. The unconditioned response would be feeling fear or pain. And because they've formed an association with being in a crowd, with that getting jostled, they now produce a conditioned response of fear. If we then remove that one step further and say, okay, well, somebody who has a fear of crowds, and it's not an irrational fear because they've experienced injury in the past, they're in an area that isn't crowded, like a movie theater, but that movie theater becomes crowded, and they start to feel fear in a movie theater. And that's because they've now come to associate the movie theater, what was a previously neutral stimulus, with a conditioned stimulus, which is crowds. And so when this happens, they can come to associate the movie theater itself, even when it's not crowded anymore, with that conditioned fear response. So this sort of one step removed is how we can have irrational fears develop. So this is a very prevalent form of learning in humans. We learn a lot about our environment this way. Our second example is the sensory preconditioning. Now this one's kind of cool, and I hadn't really thought about it until I started teaching about learning, but then once you see it, it's like, oh, that makes sense. So for sensory preconditioning, we're still going to have two conditioned stimuli, but in this case, we are going to start by associating the two conditioned stimuli with each other before we have any classical conditioning occurring. So this would be a preconditioning phase or a pre-exposure phase. So step 1 is our conditioned stimuli, CS1 and CS2, are associated with each other. And they're presented together so often that we associate CS1 with CS2. Next, we're going to learn something about one of our conditioned stimuli. So we can take CS1 and associate it with an unconditioned stimulus. So this is a normal classical conditioning exposure here. So we can get our CS1 to elicit a conditioned response because of its association with that unconditioned stimulus. However, as we learn about CS1, CS2 happens to get dragged along with it. Because we associate CS1 with CS2, CS2 comes to elicit that same conditioned response, even though it was never directly paired with our unconditioned stimulus. Now, this is kind of a weird one to wrap your head around, so let's use an example. If you think about having lasagna and having garlic toast, if you're like me, 99% of the time when you have lasagna, you're also going to have garlic toast. So whenever I think of lasagna, I think of garlic toast. Whenever I think of garlic toast, I think of lasagna. Now, let's have a conditioning trial where we have a bad batch of lasagna. Maybe something went off, maybe it wasn't cooked right, and you get sick. So our conditioned stimulus of lasagna is now associated with that unconditioned stimulus of being ill. And our conditioned response is a conditioned taste aversion, where now when we think of lasagna, we think, ew, that made me sick. As a result, now when we think of garlic toast, which never actually made us sick, now makes us think that it's gross because of that association that we learned with lasagna. So garlic toast got dragged along because of what we learned about the lasagna. All right. So that was us talking about what makes an effective conditioned and unconditioned stimulus. What takes care of or what sets up our CS and our US. Next, we can look at what determines the nature of our conditioned response. So we've already seen a large variety of responses that can become conditioned. We've looked at salivation, eye blinking, fear, locomotor approach or withdrawal, different kinds of aversions. But why does one set of responses become conditioned in one situation, but a different set of conditions or different set of responses develop in another circumstance? The first thing that we think about in this case would be that the unconditioned stimulus is obviously going to be involved in determining the nature of our conditioned response. So if your unconditioned stimulus is food, then your conditioned response is going to be salivation, because of course it is. If our unconditioned stimulus is a puff of air, then our conditioned response is going to be an eye blink, because of course it is. And many, many studies have shown that small changes in what our unconditioned stimulus is can produce changes in what conditioned response we observe. Now, how do we think this happens? The prevailing theory is the stimulus substitution model. Now, this was originally proposed by Pavlov, and it does explain quite a bit of what we see when it comes to determining what kind of conditioned response we see. So the way that this model works is that we have this, we start off with our unconditioned stimulus. So we start with food. And there is a particular connection between our unconditioned stimulus and the unconditioned response. So there's a link in our brains between food and salivation. That's going to be a pre-existing neural connection. What this model says happens is that our conditioned stimulus, it's going to sort of activate these pre-existing neural pathways that had been previously activated by our unconditioned stimulus. Basically, our CS comes in and adds on to this existing pathway. So when we go through our conditioning, when we go through our training, we introduce this new connection where the CS activates the pathway that goes from the US to the response. So we could consider our conditioned stimulus a surrogate or a substitute for the original unconditioned stimulus. Now, this kind of explains why then our conditioned response should be the same as our unconditioned response, because all we're doing is substituting our conditioned stimulus in for the unconditioned stimulus, and we're getting the exact same response. Logically, that makes sense, but as scientists, of course, we're going to want to test this theory, test this idea. And again, the standard experiment to show this process is the Jenkins and Moore study done in the 70s, where they gave pigeons different but related unconditioned stimuli that had the same conditioned stimulus. And their goal was to see if the conditioned responses would match the similar but different unconditioned stimuli that they had used. So let's look at this experiment. Our subjects are pigeons, and they specifically used a within groups design so that they could be more sure that the birds were adjusting their behavior, as opposed to one group being different from the other. And they used an auto-shaping procedure, where, as we've talked about, when a key is illuminated, when a light comes on in one of these boxes, pigeons have a tendency to peck it. Especially if that key comes on just before the delivery of an unconditioned stimulus. Now, our unconditioned stimuli were going to be food and water. So again, we hop to the next page here. Our conditioned response, if they were trained with food, is that they would peck at that response key as though they were eating. So if we look at the images here on the far right, this is when their conditioned stimulus was food. Their conditioned response is to peck at the illuminated light with an open beak, as though they were reaching for and interacting with a food pellet. In contrast, if our conditioned stimulus was water, the conditioned response would be to peck at that key as though they were drinking. They would peck much slower, their beaks are closed here, and it would often be accompanied by swallowing. So the conditioned responses here were similar, but differ depending on what the conditioned stimulus had been. Now there's a video up on our e-class that goes over this in a little bit more detail just to show you them doing this live. Okay, so that experiment tells us that, yeah, the stimulus substitution model works. They change their conditioned response based on what the conditioned stimulus, or what the unconditioned stimulus is. That's fine and dandy. However, sometimes the conditioned response doesn't match the unconditioned response. And we've already kind of seen this in action. So, for example, if we see a rat and the unconditioned stimulus is a foot shock, the unconditioned response to that unconditioned stimulus of a shock would be to jump, to sort of break contact with the ground that's causing the shock. However, if they learn a conditioned stimulus of a tone that's paired with a foot shock, the conditioned response, instead of jumping, is actually freezing or immobility. So we see more fear responses as opposed to direct response to a shock. Another example that the textbook uses is this weird setup. So it's similar to what we've seen for sign tracking experiments. And here what they did was they used another rat as a conditioned stimulus to signal the unconditioned stimulus of food. So we have our sign tracking setup here where there's a food cup. That's where the unconditioned stimulus would be delivered. And then we have a rat. This is our conditioned stimulus. And so if our conditioned response would match our unconditioned response, then we would expect behaviors associated with eating. So maybe we would expect our participant rat to bite or lick or something like that to this stimulus rat. But instead, what we end up seeing, actually, we have it written on the next page here, but what we end up seeing is that our conditioned stimulus actually elicits a social affiliative conditioned response. Basically, the participant rat will walk over and interact with and groom and sort of just be around our stimulus rat. So they're not biting, they're not licking, they're just being around, they're affiliating with the stimulus rat. So this doesn't support our stimulus substitution model. This tells us that our conditioned stimulus itself is also involved in determining what our conditioned response should be. So basically, we're saying that it's going to depend. Sometimes the conditioned response and the unconditioned response are going to match, and sometimes they will differ based on other factors. So long story short, it's a lot more complicated than what you would have initially been taught. Another variable that we should consider is the CS-US interval. Basically, how long of a gap is it between the presentation of the conditioned stimulus and the follow-up from the unconditioned stimulus? In general, a short CS-US interval is going to activate responses that are appropriate for immediately dealing with that unconditioned stimulus, whereas a long CS-US interval is going to activate responses that prepare the organism for that unconditioned stimulus over a longer span of time. Basically, a short interval is going to get us a reaction, a quick reaction, something that's going to happen immediately, and the long interval is going to give us behaviors that allow us to prepare for and long-term anticipate the arrival of that unconditioned stimulus. And so we can come back to our sexual conditioning in male quail because, again, this is something that a lot of people have studied, and we can look at the length of intervals and how it affects sexual conditioning in these quail. So the experiment that we're going to be looking at was done by Akins in 2000. Having trouble with decimals today, but basically they had male quail who were given two different CS-US intervals. So either the CS was presented 1 minute before the arrival of an unconditioned stimulus, or the CS was presented 20 minutes before the arrival of an unconditioned stimulus. They also, of course, like good scientists, included unpaired control trials, or unpaired control groups, to make sure that there wasn't anything else going on, but mostly we care about their treatment groups. As per usual, when it comes to sexual conditioning in male quail, our unconditioned stimulus would be access to a receptive female at the end of that interval. They then measured the type of conditioned response that would be seen more commonly in each of the two delayed conditioning procedures, or whether it was a long or a short interval. All right, so if we hop to our results graphs here, we're looking at, as per usual, our responding on the y-axis and then time, actually, what? Technically time, but not in the way that we usually see time. And the bottom here, we're actually indicating our two different groups. So whether they had a 1 minute CS-US interval or a 20-minute CS-US interval. And same here, 1 minute or 20 minute. And then for our types of responding, we can look at approach behavior, which would be measured in the percentage of time near our conditioned stimulus. So when they were exposed to a wooden block or a light or one of those pseudo quail models, whatever the conditioned stimulus was, for the 1 minute CS-US interval, we see that they spent a lot of time approaching and being near that conditioned stimulus. So if they knew that the unconditioned stimulus was coming soon, they would approach and stay in the area of that conditioned stimulus. Whereas if there was a longer interval, if it was a 20-minute interval, they spent very little time in the area around the conditioned stimulus. So they wouldn't go and sit next to the block next to that conditioned stimulus. They would do something else. And that something else is what the other graph is, and that's general locomotor behavior, which is basically how much are they moving around in the enclosure. And so here we can see that our unpaired control group did show that there was more locomotion in the 20-minute CSUS interval, but that's effectively a side effect of it just being a longer interval. So as more time went on, they moved around more. But we care about this because this is showing us a baseline of how much movement we would expect if there was nothing else going on. That's why I pointed out. What we care about here is the fact that our paired group, the diamonds, showed a significant increase in how much movement they were doing even over the baseline. And so in the 1 minute US or CSUS interval, we see very little moving around. That's where we were seeing that approach behavior. So they'd approach the conditioned stimulus and stay near it. Whereas in the 20-minute interval, they'd move around. They'd engage in general locomotor behavior. And this makes sense based on something that we're going to discuss in just a couple of slides. So a quick summary of what we've seen so far before we kind of bring it all together with an overarching theory. So we've talked about the fact that our unconditioned stimulus, our conditioned stimulus, and the CSUS interval all affect the conditioned response that we observe. So we saw 3 different experiments that looked at each of these. Now, we had just talked about our Pavlovian stimulus substitution model, and this diversity here is absolutely not explained by that model. So instead, we have to think about these ideas in terms of how this would be framed or function in nature. And that brings us to the behavior systems theory. Now, we've already kind of touched on this idea already, so we're bringing more ideas in together again here. So in the wild, animals have to find and eat food, they have to defend their territory, they have to avoid predators, they have to raise their young, they have all of these different things that they have to do. And we've talked about behavior systems that may have evolved to solve different problems. So we talked about search behavior in squirrels looking for nuts, where early on they would engage in general searching behavior, and then once they find the right tree, they would engage in more specific searching behavior, and then once they found an acorn, they would engage in very, very specific behavior to open that nut and eat that food. That's when we were talking about modal action patterns. But we're going to be using that same basic idea, and we can talk about different kinds of behavior systems, different sets of behaviors that go together to help solve these problems out in the wild. And the ones that we tend to care about are going to be food behavior systems, like the example of a squirrel getting into an acorn, and the sexual behavior system, which, as you can imagine, we're going to apply to our quail. So what do these different kinds of behavior systems have to do with what we just talked about looking at our sexual conditioning in quail? Well, let's pull up a doodle form here. And so we can have, early on, you're going to be seeing general behaviors, so general searching behaviors. When we talked about our squirrel example, this would be a squirrel trying to find the right kind of tree. After that, once they've found the right kind of tree, they could then engage in focal behaviors. So you're in the right area and you can exhibit some of your focal behaviors. So you go from step 1 to step 2. And then once you've found the exact thing that you're looking for, then you can engage in those consumatory behaviors. So the squirrel gets into the nut. And if we apply this to our quail, a quail out in nature would spend their general searching behaviors looking for a mate. So they look around for an area where they might find a female. That would be our general locomotion. So that's the quail walking back and forth in that enclosure when we had that longer interval. If we then have a shorter interval, it's like hopping in on this timeline. Instead of coming in back here at our general searching behaviors, we would hop in somewhere in and around here-ish, where we're seeing focal behaviors or even consumatory behaviors, which in this case would be copulation. So this is when they're spending a lot of time in close proximity to our conditioned stimulus. So this is if we have a block here, the quail would be circling this stimulus. They're spending time in that area because it's a focal behavior, because this is the shorter interval. So we looked at this in terms of quail and their sexual behavior. This also applies for squirrels and food, but those time intervals can determine where in this general system that the behaviors we're looking at are going to. The next thing we're going to talk about is how our conditioned stimulus ends up actually producing our behavior. So what kind of mechanism do we think is going on? And to do this, we're going to look at SR and SS learning. So SR learning is basically saying that a new stimulus-response connection forms between our conditioned stimulus and the conditioned response. This is different from SS learning, which lines up with Pavlov's view of stimulus substitution. So SS learning says that our CS is activating a representation of our unconditioned stimulus, just like what we talked about with stimulus substitution. So let's look at diagrams to see why these two are so different. So SR learning first, we're seeing that standard setup, our conditioned stimulus is associated with our unconditioned stimulus and produces an unconditioned response early on in conditioning. So this is during our acquisition phase. Over time, as that conditioning is happening, more and more associations between the CS and the US becomes unnecessary, and we see a direct link between our conditioned stimulus and now what is our conditioned response. So we're seeing a new connection between our stimulus, the CS, and the response, our conditioned response. Unlike for SS learning, which this is going to look very familiar because we already did this part, where you would see that CS paired with the US leads to an unconditioned response. For the SS learning, it's the same diagram where the exposure to the CS is activating our conditioned response by way of some kind of representation of the unconditioned stimulus. Basically, our conditioned stimulus is activating a quote-unquote memory of our unconditioned stimulus. So our conditioned response is basically because of this activation of that representation of the unconditioned stimulus. Now, that's all fine and dandy, but which of these is the case? How do we tell if it's SR or SS learning? The standard way to pick these apart is to use something called the US devaluation paradigm, unconditioned stimulus devaluation paradigm. What we're doing is we are devaluing or making the unconditioned stimulus less desirable or less important. So step one in a devaluation paradigm is basically standard conditioning. You're going to set up an association between our conditioned stimulus, the unconditioned stimulus, and it's going to produce a conditioned response. Phase two is our actual devaluation, where our unconditioned stimulus, which was something very big and attention-grabbing, something salient, becomes something smaller and less salient. It's less intense and it's less worthwhile. If we've reduced this unconditioned stimulus, we can then look and see what happens now to our conditioned stimulus now that the unconditioned stimulus is less meaningful. if our conditioned response is being elicited by way of some association or representation of the unconditioned stimulus, we would expect exposure to that conditioned stimulus to produce a less intense conditioned response. So if the unconditioned stimulus still matters, if we're using SS learning, then we'd see a reduced conditioned response from devaluing our unconditioned stimulus. So if we look over here, we can see kind of a reminder of what we'd be looking for. So I just talked about if it's that SS learning, then the subjects in the experimental group, this group on the top where we devalued our unconditioned stimulus, should respond at a lower level to that conditioned stimulus as compared to our control group. So if we see a reduced in the conditioned response, after devaluing our unconditioned stimulus, it's SS learning. If, however, our unconditioned stimulus has no effect on our conditioned response, if devaluing our unconditioned stimulus still allows us to see the same high level of responding, then we know that the unconditioned stimulus was unimportant, and we're more likely seeing a SR association and SR learning. So if the devaluation works, then it's SS learning, and if it doesn't work, then it is SR learning. And I had put this here in case I wanted to flip ahead, but I just flipped backwards, so not important. Let's look at an example, an actual physical example of this devaluation procedure. Now, these diagrams are from a larger experiment that looked at both first order and second order conditioning. We really only care about their first order conditioning, so we're looking at 1E and 1C. 1E meaning first order conditioning experimental group, and first order conditioning control group. Now, this was a fairly simple setup in terms of a conditioning experiment to set it up originally. So they had a tone that was associated with the delivery of food. Our conditioned responding here that we care about on this graph is basically how many times they'd interact with a lever in the cage. The lever wasn't required to get the food because we're still looking at classical conditioning. It's just sort of a byproduct of how much activity that we're seeing. And so actually, if I hop back here, so phase 1 is the conditioned stimulus is the tone, the unconditioned stimulus is the delivery of food, and the conditioned response is that they're interacting with the food or with the lever, as the case may be. Phase 2 is that they devalue our unconditioned stimulus here being food. So how do you devalue food? You make sure that the rats are full. So they gave the rats lots and lots of food prior to the next part of the experiment. So they said, okay, if the rats associate this tone with food and we give them tons of food so that they no longer want food, what happens? Do we see less responding because food doesn't matter anymore? And in fact, we do see a decrease in responding. So looking at our experimental group here, we see much less lever pressing. And over here, we also see that they ate far fewer pellets. So we're seeing less responding. We're seeing a lower conditioned response to presentations of our conditioned stimulus after we devalued our unconditioned stimulus. So this is giving us support for our SS model of learning. Now, I'm not going to talk about it here, but there is evidence for SR associations, but we have an entire section that we'll talk about that in Chapter 7 because it's a little bit more complicated. So what we're going to take home from this is that we do have evidence supporting SS learning, and we will eventually talk about evidence for SR associations as well. Now the next bunch of slides all kind of go together, so I think I'm going to stop here and we'll pick up from this point for Thursday's lecture.